Okay, so as we discussed earlier in the lecture, bacteria have different protein appendages which allow them to interact with the environment. And these include fimbriae, pili, and flagella. So fimbriae are short bristle-like fibers on the surface of bacteria and are usually present in the hundreds. They're useful for adherence to other cells, and they're also involved in biofilm formation. So biofilms are colonies of bacteria embedded in an extracellular matrix and attached to a surface. So another structure which is useful for attachment are the pili. And these are evolved in attaching bacteria to surfaces, but these are longer than the fimbriae and present in less numbers. And there's also a specific type of pilus or pili, which is involved in the transfer of DNA between bacterial cells. We also call this conjugation. And just to recap, so the difference between the fimbriae and the pili. So fimbriae are typically more numerous and shorter, whereas the pili are longer, present in less numbers. So another very important appendage is the flagellum, which is a singular, or flagella plural. And these flagella have a long whip-like structure for the bacterial motility or movement. And these flagella act like propellers, and they propel the bacteria through the aqueous environment. So there are basically 4 different types of flagella arrangements. So the bacterium can just have one flagellum at one end of the cell. So this is called a monotrichous flagellum. Or the cells could have flagellum at each end, called amphitrichous flagella. Or the flagella can be present as a tuft on one end. So multiple flagella can be present at one end of the cell, and that's lophotrichous flagella. Or the flagella could cover the entire surface of the bacteria, and in this case they're called peritrichous flagella. So flagellated bacteria are able to undergo directed movement through changes in the rotation of the flagellum. So when the flagella rotate counterclockwise, they form a large bundle which propels the bacterium along a straight line in a single direction. And this is what we call a run. Okay, so that's counterclockwise direction resulted in a run. But when the flagella rotate clockwise, this causes the bundle of flagella to disassemble and the bacteria tumble randomly in no particular direction. So when no chemical gradient exists, the lengths of runs and tumbles are equal and the overall movement is random. So there's no overall directional movement. However, when there's a chemical gradient of an attractant, the length of runs extends while the length of tumbles decreases. Okay, so this next slide looks at binary fission in bacteria, and this is how bacteria reproduce, and this is by asexual reproduction. And by asexual reproduction, we mean reproduction which does not involve fusion of gametes. So this is unlike humans where the sperm fertilizes the egg, and you've got this fusion of gametes. So with bacteria, first of all, you get the replication of the chromosome, and then the chromosomes separate. Septum or dividing wall then forms down the middle of the cell and splits the cell down the middle, and then 2 identical daughters are formed, each with a chromosome copy. So these daughter cells are identical to the parent. So we mentioned gram staining very briefly early on. So gram staining is used to differentiate between the gram-positive bacteria and the gram-negative bacteria based on their different cell wall components, as we discussed. So the first step in the process of gram staining is to add the primary stain, that's the crystal violet, to the cells. And this stains all the cells purple, so both the gram-positive and the gram-negative purple. Then the second step is to add the iodine solution, and the cells remain purple at this point. Then the third step is to add the alcohol, which washes away the stain from the gram-negative cell walls. However, the gram-positive cells remain purple when the gram-negative cells become colourless. And then the final step is to add the safranin, which allows dye to adhere to the gram-negative cells. So therefore you have the gram-positive cells which have remained purple all along, and the gram-negative cells now appear like a pinky red because they've been stained by the safranin. And the difference in the staining between the gram-negative and gram-positive is due to the difference in the thickness of the peptidoglycan layer in the cell wall. So as we mentioned, gram-positive cell wall has very thick layers of peptidoglycan, which actually resists the decolorization step in the staining, which enables the gram-positive cells to keep their purple stain, whereas the violet stain is washed out of the gram-negative bacteria due to their thinner cell walls. So simple staining of bacteria is just a quick and easy way to determine cell size, cell arrangement, and the shape of the bacteria. So simple staining is just as it describes, very simple staining procedure that uses just a single solution of stain. So for example, a basic dye such as methylene blue, safranin, or crystal violet can be used to colour the bacterial cells. These stains will readily give up a hydroxide ion or accept a hydrogen ion, and this leaves the stain positively charged. And since the surface of most bacterial cells, the cytoplasm, is negatively charged, these positively charged stains adhere readily to the cell surface. So this simple staining is not great for differentiating between bacteria or differentiating between the different components of the cell. So differential staining techniques, on the other hand, such as gram stain and acid-fast stain, are used more often than simple stains as a means of gathering information about bacteria. Differential staining methods usually require more than one stain and involve several steps, but these are actually better for differentiating between different types of bacterial cells or between different parts within the bacterial cell. Okay, so we've come to the end of the lecture, and in summary, you've learned about the major components of a bacterial cell and how these are important for survival. We've also learned about specialized bacterial appendages, such as flagella, PM1A, and pili, and also how they're involved in movement and attachment of bacteria to different surfaces, sites. We've also learned about the sporulation process and why spores are so vital for the survival of certain bacteria, and also why they're so hard to destroy. And we've also learned about different staining methods used to identify and characterize bacteria, including gram staining in more detail. And just so you can test yourself on your knowledge, I'll be putting up a recap activity for next week. So you can test yourself on everything you've learned, see if you remembered it, and you can also apply your knowledge to some different scenarios. Okay, thank you, and speak to you soon.